Hi everyone, thanks for coming this afternoon. So I'm just going to give a quick intro. So this afternoon we're going to have 3 talks. I'm not sure if many of you were here in the morning, but we had 2 talks then. So this afternoon might be a little bit longer. So just to start off, if everyone could keep their microphones and cameras off, unless at the end of a talk you want to ask a question or anything, you can turn your microphone on or we can turn it on. And if you have any questions at all throughout a talk, feel free to type them in the chat and then we can go through them in the Q&A afterwards. Perhaps if you could go to the next slide, please. Yep, so this is just the summary of what we're going to be listening to this afternoon. And at the end, if any of you are not University of Leicester students and you want to ask any questions about life at university, or maybe what you might want us to do, like ChemSoc to do, any events or anything, feel free to hang around and we'll answer any questions and talk about that. If you go to the next slide as well, please. Yeah, so also let us know if you can't hear us or if you can't see any of the speakers or presentations, and we'll do our best to solve all of that. So I'm just gonna spend a few minutes now just telling everyone about some of the events that we have planned this year. So the first one that we're hopefully going to do is a pub quiz, and we're hopefully going to get some of the lecturers to come along to make things a bit more interesting. So ideally the first week, so Freshers Week, so that will give a nice welcome to the freshers, and obviously it'd be quite nice to have a proper in-person pub event that we haven't managed to have in the past year. And then of course we're going to aim to do a spring ball, all COVID permitting. I've copied a link which I will just paste in the chat if I can. This is just a Google Docs form that ChemSoc have made, and it's just to ask if there are any events that you might want us to do or any suggestions, so feel free to fill it out. Does anyone have any questions at the moment about how this afternoon is going to run or anything like that? I'll take the long pause as a no. Okay, so first up we're going to have Fab speaking to us, and he's going to be talking about his research in sustainable synthesis and catalysis.
Thanks, Rosie. I'm just going to share my screen now. 1 second. Okay. Can you see the screen?
Yeah, we can see it.
Excellent. Okay. Right. Okay. So thanks for being here today. Whoever came to the morning session, or thanks for making it to the afternoon session, I guess. So yeah, today I'll talk about some of the research that my research team does in sustainable chemistry. But also I will give an overview of some of the research done by the Sustainable Synthesis and Catalysis group here at Leicester. So it'll be a bit of everything really that we do. So first of all, why sustainable synthesis and what do we mean by sustainability? So it's a very broad definition, but myself, I'm an inorganic chemist. So I focus very much on the role of metals in sustainable synthesis, and it all boils down really to this modified version of the periodic table that was published by the Royal Society. I think it was UChemS actually. Well, a few years back. So it's color coded, and it just tells you essentially. So green is the color for the most abundant elements, the ones that are most readily available. When you go towards the red, instead we go towards the metals that are less available, and they're considered critical materials. So in principle, when it comes to industrial processes of things that really sustain our way of life, we don't really discriminate between materials that are less available or more available. We're just trying to make things work. And it's been like that for a very long time throughout the 20th century. Well, since the start of the Industrial Revolution, really. But this has changed quite a lot over the last 20 years. There's a more of an understanding of the demands of industrial processes on the environment, on the earth, on what we do to our planet. So we're trying to make industry more sustainable and trying to be kinder essentially to the environment. We still need these industrial processes. We cannot just shut down our industrial plants and then pretend that we can sustain our way of life without certain industrial processes, because we need them for making drugs, pharmaceuticals in general, for making pesticides, for making fuels that power the jets that fly across the continent. Well, when we were allowed to fly pre-COVID, I guess. And many of these processes, though, rely on critical materials, particularly metals that are around here, so very nice red and orange colors, so particularly on palladium, platinum. These are two of the metals that are mostly used by the chemical industry in a lot of these processes. And there is a demand problem. So the supply, so the demand is vastly higher than the demand. So if you can see from this graph here, if I switch to laser, oops, laser pointer. So this is roughly the demand that we have right now for palladium. And you can see the supply is far lower. But we obviously recycle some of these metals. There are processes which can re-extract these metals, for example, from catalysts used in cars. But still, the demand is still superior, or we just almost about break even. The other problem, obviously, these metals are quite scarce. They're difficult to extract. And this is reflected in the cost. So you can see the cost per kilogram is absolutely enormous. So in an ideal world, to make processes more sustainable, at least processes that use these precious metals, we should use earth-abundant cheap metals. And this is where my research interest really comes in. So in our research group, we study metals that are relatively abundant in the Earth's crust. So two of them are magnesium and calcium, which are, if I remember correctly, the third and fifth most abundant elements on the Earth's crust, or thereabout anyway. But you can see the abundance is orders of magnitude superior to palladium and platinum. Another interesting topic in our group is also looking at the so-called F element or rare earths. And although they're called rare earths, these metals are actually not that rare. So these are lanthanum and xenon, the two most abundant rare earths, and you can see their abundance is still much, much higher than that of precious metals. Even the least abundant rare earth, thulium, is a lot more abundant than palladium platinum, and it's just as abundant as, well, just a bit more than iodine. The common theme about all these metals, they are produced on very, very large scales. Although the extraction of the rare earths is not particularly environmentally friendly, there's a lot of them spread around the world in various bits of technology, which I'm going to talk about in a minute. So the big scientific question that we pose is, can we replace precious metals, very expensive metals, in chemical synthesis so that we can improve the sustainability of our industry? Well, the answer is maybe, potentially, we can do it. So if we identify metals that are cheap and can then have a very positive impact on the both economy and the environment and the environmental impact of these processes, then we could look at introducing them in these new processes. However, there's a lot of ground, there's a lot of stuff that we need to do first. First, we need to understand the fundamental properties of these metals, and maybe find properties that we still don't know at present that might actually make these metals suitable alternatives turn into suitable alternatives for precious metals. So we start really with a fundamental, hardcore fundamental science before we can go to the applications. And this is very much what we do in my research group. So for those of you who don't know, so the group 2 metals are here. So they go from beryllium to radium. And I'm not really going to talk much about beryllium and radium today. So beryllium obviously is a very toxic metal, so there's no lot of interest in the applications in the industry. We briefly talked about this in the early this morning with Carl Ryder, if you were there. And when it comes to radium, instead, it's highly radioactive. So very unlikely that anyone would want to put radium in a, I don't know, in a car, for example, in an autocatalyst. Lanthanides and actinides, they constitute the so-called F block. We don't do any research with the actinides here at Leicester, but we do look at the lanthanides quite a lot. And they are so separate from the periodic table. They are a bit of the altogether the black sheep of the periodic table. They have their own very unique properties. They're less well studied, and I guess that's why I really like them, because no one, well, for a long time, people don't really look at them in the same way they look at transition metals. So alkaline earth chemistry is extremely well established. So it goes back nearly 150 years. It's mostly renowned for the work of Victor Grignard at the beginning of the 20th century. And so he developed what are known now as Grignard reagents, which are organomagnesium compounds where we have magnesium bound to a carbon, a highly nucleophilic carbon. And what these reagents do, they generate, they're used to make new carbon-carbon bonds. So we have a new nucleophilic center that can attack unsaturated substrates. And this is used quite a lot in synthesis. In fact, if you do any undergraduate degree in chemistry, you will end up making Grignard reagents in teaching labs at some point, sometimes in first year and second year. If you then become a researcher, or you do any bit of synthesis, even high-level synthesis in let's say in a drug discovery environment, you will certainly be using Grignard reactions at some point. Really, it is one of the fundamental pillars of modern synthesis. And they've essentially dominated group 2 chemistry for the best part of the 20th century. The chemistry with the heavier elements wasn't as well developed for a long time, but this has changed slightly over the last 20 years, particularly to the work, thanks to the work of people like Mike Lappert, Matthias Westerhausen, and more recently, Cameron Jones and Rob Gillard. So what now these people have done, they started expanding, they expanded the fundamental knowledge of the other elements of the group 2, particularly calcium, strontium, and in the case of Rob Gillard, a bit of beryllium chemistry as well. And there's not many people in the world that do beryllium chemistry because of how toxic it is. So things are changing, and there's a lot of scope for new chemistry to be developed. With regards to F-element chemistry, so lanthanides, they are applied in synthesis, but not, the applications are not as broad as for other transition metals or even for the group 2 metals, but they used a lot in consumers technology because they have remarkable physical properties. So they're using lasers, magnetic devices. In fact, today you're attending this virtual event, and probably if you're using a computer, there'll be an awful lot of rare earths or lanthanides inside the computer, starting from the neodymium magnet in your hard drive to the technology that's behind the screen that you're using to look at me right now. So they're really ubiquitous in our modern society. And actinides are also quite popular for very different reasons. They play a big role in the nuclear industry, obviously. So in the energy sector, and unfortunately, they're renowned for the military applications, as you might well know. But behind all these applications for lanthanides and actinides, there is still the fundamental science underpinning them. So everything started from developing fundamental concepts that then could be translated into new technology. And there is a lot of coordination organometallic chemistry behind it, which is what I'm very much interested in. So going back to some basic properties of the metals. So for those of you who are doing first and second year undergraduate studies, you should be quite familiar with this picture. So this is, as in a nutshell, transition metal coordination chemistry summed up in one slide. So that's almost all the things you need to know. Not all of them. It won't be enough to pass an exam, but it would be a good start point. So the coordination chemistry is fairly predictable when it comes to transition metals. That means that when we put a metal with a certain ligand environment, we should be able to tell the shape of the molecule that is going to come out. Coordination numbers vary from 2 up to 6 or 7. And the most common shapes are, for example, the tetrahedral shape, the square planar, or the octahedral shape. And a lot of the properties are linked to these pretty pictures here. So these are the d orbitals, which are simple representations of the electronic distribution around the nucleus. So this gives us the probability of where the electron is going to stay at any one point in time. And there's a strong correlation between the physical-chemical properties of the transition metal complexes and these d orbitals. Another interesting properties of the transition metals is that they have multiple oxidation states readily available. So they can flick between, they can gain electrons, they can lose electrons reversibly, that's what makes them very palatable for industrial applications. So this changes a lot when it comes to alkaline earth and lanthanides. So with the exception of beryllium and magnesium, the alkaline earth metals are very, very big, much bigger than the usual transition metals used in synthesis. That's the same for the lanthanides. They have very large ionic radii, and the coordination numbers, instead of stopping at 6 or 7, they can go as high as 9, although there are also some compounds that have coordination numbers up to 12. The geometric properties of the resulting compounds are a lot more complicated, a lot more difficult to predict. And in the case of the lanthanides, when we said that the orbitals, they give us a representation, or well, they give us a tool that we can use to predict properties for the transition metals, the equivalent for the lanthanides are the f orbitals. These rather complicated but quite fascinating pictures here. So once again, these are electronic distributions or probability of finding the electrons around the nucleus, and they are an awful lot more complicated. So a lot more nodal planes. So as a result, the properties are probably, you could have a guess, a lot more complicated to predict. In fact, when it goes down to pure theoretical chemistry, it's a lot more computationally expensive to study these elements. A common theme between the alkaline earth and the lanthanides is that the redox chemistry is limited. Group 2 metals, they are normally closed shell. They don't really, so once they go from metallic state to the +2, so when they lose 2 electrons, it's very hard to make them go back. And also they don't like to go in a state in between the 0 and +2 state. Lanthanides, instead, they are mostly limited to the +3 oxidation state. So since the redox chemistry is so limited, the applications in catalysis, which makes a great use of changes in oxidation state, is limited as well. So since these metals have such a large ionic radii, when we want to design compounds, so when we want to put ligands around the metal center, we need to use very judicious ligand design. And if we are able to put together a ligand that fits the metal quite in the correct way, so leaves an open space for a substrate to come in, is protecting it from undesired substrates, then we can unlock some new properties. And when I try to explain ligand design to my students, the best analogy I could come up with was that of an umbrella. So at the end of the day, we need to we to think of the metal as something that we need to protect from the outside environment. So we want to design an umbrella that can open around it, wrap around it, but also needs to be at the right size. So if we put a metal any way down here, the stick, we want to make sure that it's the protection that the umbrella has around it won't prevent it from, let's say, getting water not just from the top, but also from the side a bit. So, but if we make a metal way too big, then we might make it stop interacting with anything else, which is quite undesirable when we want to make our complex interact with a substrate. So this, in terms of ligand designing, means is normally referred to as steric pocket. So the ligand has a very snug space where the metal will fit. So we want to make sure that the metal size and the ligand steric pocket match. And they also need to match for the properties that we want to unlock. Just to have an idea, to give an idea of the applications that of coordination chemistry and ligand design, one of the most important ones for me is the use of the development of separation strategies in the nuclear fuel cycle. So once we put nuclear fuel into the nuclear fuel cycle, this is obviously this is first enriched, then it goes into the reactor where it's burned, so to speak. What comes out is a horrible mixture, and this mixture can either be buried on the ground straight away if you want, or we could try and find a way to reprocess it and either store it more safely or do something with it, maybe put it back into the reactor as a reprocessed fuel. Now, this is not easy because when the fuel is burned, what comes out is an absolutely horrendous mixture that contains all sorts of things, but crucially contains a lot of lanthanides and a lot of actinides. So if we find a way of separating these two species, then we have a first good separation, because lanthanides and actinides are fairly similar in terms of coordination chemistry. So once we are able to separate these two, then it should be fairly easy to separate them from all the rest. That is the mess of in spent nuclear fuel. And it all boils down to this picture, which might be a bit obscure for people now in the field, but what this is telling us is the radial extension of the orbitals. So what this is telling us really is that those electronic distributions that I showed earlier, those the F orbitals, in the case of the lanthanides, are more contract, so they're a lot closer to the nucleus. When it comes to the actinides, they're a lot further away, which means that lanthanides and actinides, when they use their orbitals to interact with the ligands, they interact differently, simply because of the radial extension. And we can design our ligands so they can interact worse or better with the lanthanides and actinides and separate them. And this is something that the nuclear industry does. It's called the PUREX process. And it's used for extracting plutonium and uranium from spent nuclear fuel. Once these uranium plutonium are extracted, they can be then reintroduced in reactors that they use, these so-called MOX fuels, although there's a big controversy on where the industry is going, because we were supposed to recycle all the plutonium that we have in the country, and we haven't been doing it. And in the UK, we're actually storing plutonium that comes from all over the world. We even have plutonium that comes from Fukushima. It's all stored up in Cumbria. So, but if we can separate them, then we can create these MOX fuels. And this process, what it does is applying coordination chemistry. We're actually making real coordination compounds that have unique properties depending on whether we're binding an actinide and a lanthanide. And we use the differences in the redox properties of these complexes to separate them. So this is a generic distribution of species in nuclear fuel, in spent nuclear fuel. See, we have obviously some uranium left, there's a little bit of plutonium, there's some other actinides. There's a whole bunch of stuff like group 1, group 2 transition metals, but there's an awful lot of gadolinium, so one lanthanide. And this is the main target of this separation process. So being able to separate, get rid of these lanthanides. And it works very well. And it's just one example in which coordination chemistry is solving a major real world problem. So glad to see that actually it serves something. So with regards to my research group, we don't really look at lanthanide chemistry. As I said, here in Leicester, I used to do that during my postdoc years in Manchester. What we look at instead is lanthanide and alkaline earth chemistry, and we have quite a few different research topics here, such as the use of reagents for CH activation chemistry. Also, we are developing new organometallic reagents that can be used in catalysis. We're also looking at alkaline earth lanthanide metals in unusual oxidation states. As I said, the redox chemistry of lanthanides and group 2 metals is quite limited. So we are trying to challenge that assumption. And we're also very interested in just structural chemistry in general and getting nice, pretty pictures of compounds. I'm going to focus on these two aspects just for the next few minutes. So alkaline earth chemistry, so organometallic chemistry and small molecule activation chemistry. So first of all, how do we do what we do? So the compounds that we make in our lab are extremely air and water sensitive. In fact, if they are exposed to air and water, sometimes they can burst into flames. So this requires very specialized, highly specialized equipment. The main tool that we use is this thing here. I don't know if you can see it. It's called Schlenkline. It's a dual glass manifold. One part is connected to a vacuum line here, a vacuum pump, and the top part is connected to a high purity gas line. In fact, the gas has to be, it's an inert gas, normally nitrogen or argon, and it has to be so pure that we actually need to pass it through some scrubber that will get rid of any water or oxygen. And this scrubber actually reduces the level of oxygen down to less than 1 parts per million. So that's how pure it has to be. If we have oxygen in quantities any higher than that, our chemistry simply won't work. Then we handle all our solids in these globe boxes, which are just large boxes full of inert gas. Once again, where you can go inside with those gloves and then essentially work as if you were on the bench, although it's not as comfortable. And ultimately, we want to make crystals and put them in an X-ray diffractometer and get a pretty picture out of our compound. Make it sound easy, it really isn't. And there's a lot more that we do in terms of techniques as well. So this is one of the projects that I described earlier. So we developed new alkaline earth lanthanide organometallics. This is a project led by one of the PhDs in our group, Matthew Stevens. And he's worked alongside BSc students, MChem 4th-year students in our labs, and very much applying also concepts that we cover in some of our taught modules, particularly the one that I teach, which is CH3205. What Matthew has been doing is developing these heterolytic organometallic complexes that are used in hydroelementation catalysis with a particular focus on hydrophosphonation catalysis, so making new carbon-phosphorus bonds. What you get at the end is our new phosphines. And phosphines are very important because they are used for organocatalysis, they're used for asymmetric catalysis. In fact, many of the drugs that we use nowadays are made through some kind of asymmetric catalysis at some point. And most of the time, the ligands used in those reactions are phosphines. So there's a big demand for new phosphines or very tailored phosphine for certain synthetic protocols. That's just one of the applications they have. So another project in our group looks at subvalent alkaline earth chemistry. So where we have group 2 in unusual oxidation states. So between, rather than being +2 or 0, we're looking at +1 alkaline earth metals. This project is led by Alex Bowles, who is a first-year PhD student. And he was actually an NChem in our lab. And that's how this project started. It started as a final-year NChem project. And this type of chemistry is quite important for us because we are looking at how we can activate simple molecules like carbon monoxide and hydrogen to make complex hydrocarbons using these subvalent species. To give an idea how important this is, the the Fischer-Tropsch process, which is used for doing the similar reactivity. Well, in the UK, we're building 20 of these plants, which by 2030 will supply all the liquid fuel in the country. So there's a lot more in the synthesis and catalysis group that we do. So this is a brief overview of Dr. Alison Stewart's research. She looks at the incorporation of fluorine into molecules because most of the a lot of pharmaceuticals do contain fluorine. Actually, there's around 200 pharmaceuticals now that contain fluorine. So she finds a new and more sustainable ways to do this. And they developed this fluorinating agent, which is actually able to fluorinate target molecules very, very selectively. And so she's building essentially new building blocks for new drug candidates. And our research topic developed in a subgroup is the one by the Sundaraligan group, where they look at the design of new inorganic complexes that could be used in anti-cancer therapy. And what they do, they make compounds just like in my group, so the new inorganic complexes, and then they test them for the cytotoxicity, and then they also test them eventually in animal models. So by working in this group, you'll be at the interface between chemistry and synthetic chemistry and biology. Then we also have the group of Sandy Kilpatrick. He's very interested in organometallic synthesis, like myself, and the application of sustainable catalysis inspired by nature. So he looks at the enzymes and how the reactive sites can inspire new transitional metal complexes that then can be used for making new polymers, new fuels, and new energy materials. And finally, if you want to find out more, there's also other groups in our synthesis and catalysis group, like the group of Alex Pulis and Greg Solan. So just visit our research website to find out more if you want. So just one final slide for me. I'll just on time. So should thank really the people doing the work in my group. Really, a great bunch of people. This is the last picture. Unfortunately, it was virtual because of the pandemic. And we have a great group of collaborators between the UK and also in the US. And we're very thankful for the support that we received, even though we're a very young research group, but being quite lucky with that. And if at any point you want to know more about research, I'm happy to answer emails or just have an informal chat anytime. Okay, thanks.
Thank you for presenting. Does anyone have any questions at all? Just something in case people are typing into the chat. I was just wondering whether there are any companies that you aim to work with in the future, or if there are any like on the horizon.
So it's a bit of a difficult question, to be honest, because yes, there are and there aren't. So there's several companies in the UK that are very interested in they're very interested in looking at new technology like ours, like for example Johnson Matthey, but at the moment they're more focusing on recycling critical materials like palladium and platinum, for example. That's where the main focus is. But there's a network of companies. The acronym is called KTN, and I cannot remember what it stands for. And it's a network that you engage with whenever you're developing something like our group is doing. So there are some potential partners, but what you need to show them first is the fact that this is actually feasible. Yeah. So before you can actually go to a scale up, you need to show that the economics work. Otherwise, they won't just take a new reaction and then say, "Oh yeah, we'll take it." We need to show that you can get there. And there's actually good opportunities here in Leicester because, for example, in the material center, there's actually a pilot plant where you can try to do small scale up version of your processes. So if you want to approach a partner, you can actually show, we can do the tests here, which is one of the benefits of doing research here in Leicester, really.
Okay, cool. I think we have a question in the chat.
So on average, how many synthetic steps to make metal complexes? The main challenges of their synthesis. Yeah, thanks, James. So I think, so it depends. Sometimes the ligand can be the thing that takes the longest to synthesize, although we try to keep it as streamlined as possible. So of the compounds they are shown today, probably looking at maybe 3-4 steps in total between from making the ligand to the final complex. The challenges are the handling. So the fact that these compounds are very water sensitive. So any mistake, any error in the technique will result in like wasting weeks of work, like quite easily, which is also a challenge for the applications in the industry, in the industrial environment, because I guess the first step is to make them and prove you can make them, and then try and find ways to make them actually manageable from in an industrial setting. So the next step in our research would be to make them less and water sensitive. Otherwise, no one will want to use them. Okay.
Do we have any more questions at all, or any comments anybody might like to make?
Everyone's happy.
Okay.
Okay.
I'll take the silence and no messages as everyone is ready to move on. So thank you very much for speaking with us today. This was Dr. Ortu. Okay, so up next we have Dr. James Hodgkinson, and he is going to be talking to us about his research in chemical biology.
Thanks, Rosie. Could you just start? Can everybody hear me okay? Is the speaker okay?
Yeah, we can hear you.
Yeah.
Brilliant, brilliant. Right, I'm just going to try and share my slides now. Okay, can everybody see that?
Yeah, we can see your slides.
Okay, so apologies, I'm not going to put my video up, and the reason for that is my bandwidth isn't that great, so I just want to try and make sure there's no interruptions really in the slides. So as long as you can see the slides and hear me, hopefully that's fine. And so Dr. Ortu has just given you an introduction into synthesis and catalysis, one of the main research areas in the School of Chemistry. And, you know, I'm going to talk to you today for roughly 25 minutes about chemical biology, which is another research area within the School of Chemistry. The intention of my talk really is not so much to talk about my research as such. There will be a little bit about my research, but my intention mainly is to give you an introduction to chemical biology, what it is as a research area. I know some of you are undergraduates, maybe looking to do an MChem subject or a BSc project, and you might be thinking about the research area that you're interested in coming up. So hopefully this will give you an idea of what chemical biology is. Some of you may be prospective undergraduates, and some of you may even be looking to do PhDs in the future. So hopefully, you know, after this talk, you should have an idea of what chemical biology is. Okay, so what is chemical biology research? I've just kept this in its broadest terms. So most of this information you can find on the University of Leicester and the School of Chemistry websites about the research areas. And I've particularly picked them because afterwards, if you want to research chemical biology area more in your own time for future projects, you know, this should give you a good go-to. So the first thing I'd say about chemical biology is it's multidisciplinary. You know, we have medicinal chemists working together, synthetic organic chemists, inorganic chemists, biophysicists, including physical chemists. We all work together in chemical biology. So the first thing I'd say is it's very a multidisciplinary subject. And we have combined expertise in organic synthesis, and primarily I'm a synthetic organic chemist who works in chemical biology, and also obviously physical measurements as well. Spectroscopy plays a big role in chemical biology too. And I guess the big thing about chemical biology is we're trying to understand the interactions between macromolecules and complexes, mostly proteins, but not always proteins, and how they interact with one another and are used in biology, particularly in disease. We use chemistry to try and modulate molecular function of those biomolecules. And it is very much translational research. You know, a number of the molecules I've made in my lab, you know, have activity against colon cancer cells, and we've patented those molecules because they could have commercial value in the pharmaceutical industry. But not only making molecules to cure disease, there's also developing techniques that can be used as assays to look at biomolecules and how they function, can also be useful for biology. So that's from our University of Leicester, and this is just from Wikipedia. Everyone uses, you know, chemical biology. You know, it's a discipline of chemistry and biology. And really, I think this is a good description. It involves the application of chemical techniques and analysis. So whether that be making a small molecule or using a spectroscopy technique to study biological systems. And the big difference between chemical biology and biochemistry, which is maybe can be difficult to interpret, particularly if you're new to the area. You know, biochemistry is the study of the chemistry of biomolecules and regulation of biochemical pathways. In chemical biology, it's chemistry applied to biology. Yeah, so we are all chemists, and we all do chemistry, but basically we apply our chemistry to biological problems and biological hypotheses. We are interested in synthesis and on other aspects of chemistry, but as Dr. Ortu was saying, you know, they try to improve synthesis and make it more cost effective and more economical and more efficient in synthesis and catalysis. I mean, we are interested in that, but not as much. We're more interested in making molecules to study biology. So we do synthesis and other spectroscopic techniques to look at biology, but the end goal is to study the biology. Really what we're interested in is how we can use our molecules and our chemical techniques and spectroscopy techniques to study biology rather than the synthesis. So the research topics in chemical biology, bioactive molecules and chemical probes is one, and that's a particular area that I work in and I'm excited about. Basically, a lot of these are small organic molecules, so carbon, hydrogen, nitrogen, oxygen, if you remember your organic chemistry, to make drug-like molecules to interact with proteins that can be involved in disease, or quite often studying proteins with small molecules that we don't know the function. Biocatalysis, so how enzymes function and what they do, what's the important transition states in the enzyme, how they use their substrates, often small chemicals, to do their biocatalysis. Bioinorganic chemistry, so again, it's quite broad as well. They could be looking at the metals in the enzymes and how those metals catalyze chemical reactions, or it could be looking at molecules like cisplatin, inorganic compounds that also have anti-cancer activity. Diagnostics and imaging. So you may have heard a little bit about some molecular imprinted polymers in the morning. You know, these can be used for analyticals and diagnostics. In terms of imaging, you know, we can make all sorts of bioactive molecules with fluorophores and proteins conjugated to fluorophores, and that's really important. That means we can take a microscope and look in the cell at where the molecule is going into the cell or where the protein's going, and we can actually visualize biology taking place using our chemical techniques. Single molecule biology and biophysics, so that's more in the physical side. So physical chemists' involvement in chemical biology, using spectroscopy to look at individual molecules and proteins and how they function. And chemical biology here at Leicester is very, very well funded and researched. There's a Leicester Institute of Structural and Chemical Biology, mainly based in the Henry Wellcome building, where we have a cryo-electron microscope and other techniques to do chemical biology. But it has a big emphasis here in Leicester, and a number of the people who carry out chemical biology in chemistry are a member of the Leicester Institute of Structural and Chemical Biology, often abbreviated as LISCB. So those are not the only research topics. You know, sustainable synthesis and catalysis is very much related to chemical biology. A lot of organic molecules and inorganic molecules are biologically active, and we have to have sustainable synthesis ways to make these molecules. I mean, the guys who start in my lab, MChem students, PhD students, or BSc students, they always start with some synthesis. There's always a molecule to make. Again, spectroscopy is important for chemical biology. These different spectroscopy techniques for looking at biomolecules. Materials and interfaces is also important as well. We can think of antibacterial polymers that can be important in things like catheters and other biological applications in medicine. So these are the main topics in chemical biology. Of course, we're not limited to these topics, but it's all chemistry that is applied to biological problems and biological systems. I mean, these are some of the active staff members in chemical biology. So if you're a prospective student or future BSc or MChem project, these are these are staff members that you could look up their websites and see what type of chemical biology they're doing. Of course, it's not limited to these staff. You may find other staff that are also doing projects related to chemical biology, but these staff members here are definitely primarily doing chemical biology. But there will be others, I'm sure, that maybe aren't listed here involved in chemical biology. So if you're interested, have a look in the School of Chemistry website at the staff and the different research they've done. What I'm going to do now is just give a few research highlights, a few examples of chemical biology in research. And I'm not going to talk about everyone here because it's too difficult to talk about everyone, but I'd like to give you a few snapshots and just a little bit of taster of what it's like to do chemical biology research. So my name's, so I probably didn't say that at the start, but my name's Dr. Hodgkinson, or James. You know, I'm a synthetic organic chemist by training, but I make molecules to study biology and study biological processes. And really what we would really like to do is make molecules that can be used as chemical probes to study new enzymes or new proteins that could be used by members of the chemical biology community and biology communities. And really the big one is hopefully that the chemical probes that are synthesized in my lab could potentially be used to study disease. And really the holy grail is to aid drug discovery. We have made molecules that kill cancer cells. And obviously these could have important implications in drug discovery. So we're a synthetic organic chemist group, but we make molecules to study biology. We start with a biological hypothesis about an enzyme, and we think about the types of molecules that we can make to study those enzymes. So this is one type of enzyme that a lot of my research has been about in the previous couple of years. So your DNA, I'm sure you all know what DNA is made of, and you've all covered it, but you know, deoxyribose nucleic acid, you've got your nucleobase, phosphate, and sugar, all connected in a polymeric chain. But DNA is not linear. You know, it's not a linear chain. It's wrapped up with lots of other proteins, and these proteins are called histone proteins. And it's wrapped up in them to make it more compact into structures called chromatin and chromosomes as well. And you know, how the DNA is packed is really important in what genes get turned on and off. So which genes undergo transcription, and that can be important in cancer or other diseases. if a gene that's involved in cancer is turned on, that's obviously not a good thing, called an oncogene. And so what these molecules do, these enzymes, we have enzymes called histone acetylase transferase. So don't get too worried about that. Basically what they do, this is the lysine amino acid. And these molecules, histone acetylase transferases, will have the free amine here, and that will put an acetyl group on there. So a very simple chemical transformation carried out by this enzyme, histone acetylase transferase. It sees a lysine residue and puts an acetyl group onto that lysine residue. We also have enzymes called histone deacetylase enzymes, and they catalyze the reverse reaction. So they take a acetyl group on a lysine residue, and they remove that acetyl group. And the HAT is histone acetylase transferase, HDAC is histone deacetylase enzyme. And why is that important? Well, that's important because it controls the transcription of DNA. Whenever this lysine residue in this histone protein is not acylated, it has a positive charge. And of course, you know that DNA has a phosphate backbone that's negatively charged. If that's negatively charged, the DNA is going to be tightly packed around these histone proteins such that gene expression can't take place. And that's what we call a tightly packed DNA chromatin, sometimes called heterochromatin. Now, if the acetyl group is on there, it's less packed DNA. So we've lost that positive charge there. We still have the negative charge of the phosphate backbone, but we've lost the positive charge. That means this DNA is not going to be tightly packed to the histone proteins. It's going to be, we sometimes call euchromatin. And this is going to, if it's less tightly packed, it's more accessible. The DNA is more accessible to undergo transcription and gene expression. And that could be problematic if it's a protein involved in cancer. So if we inhibit a HDAC enzyme or degrade a HDAC enzyme, we can increase lysine acetylation markers in histones and chromatin. So these are 3 HDAC enzymes. There are 18 HDAC enzymes in the cell. 11 of those contain zinc in the catalytic active site. The enzymes we were interested in are HDAC 1, 2, and 3. So this is the structures of HDAC 1, 2, and 3. And you can see these little cavities just here of HDAC 1, 2, and 3. And those cavities are very simply where the lysine acetyl group goes in there and undergoes its deacetylation reaction. So these are proteins, and these are very important proteins, these HDACs. They're expressed in the nucleus of the cell and on all human tissues. So there are HDAC drugs in the clinic. What happens is if you inhibit HDAC enzymes, you can induce apoptosis, cell death, and these molecules of HDAC inhibitors have been developed as anti-cancer drugs. So one example here is furinostat. It's an organic molecule that inhibits the HDAC enzyme. Romidipsin, approved in 2009 for its uses as well, also acts as a zinc chelator. This disulfide bond is cleaved. This is interesting. This is a natural product isolated from gram-negative bacteria. So this natural product from gram-negative bacteria is actually a drug, a HDAC inhibitor used in cancer, particularly blood cancers. Belinostat, which was approved in 2014, panobinostat, which is approved in 2015. And most of these have a very similar structure in that they have a group that chelates zinc. So this is the hydroxymic acid group that chelates zinc. This is the hydroxymic acid group that chelates zinc here, hydroxymic acid group that chelates zinc here. And here, rather than the hydroxymic acid group, we have the disulfide bridge that gets cleaved and chelates zinc. And what that does, this is the structure of that protein with furinostat in its catalytic active site that stops lysine acetylation. Now, these drugs are good. They're used in the clinic, but there is one problem. They inhibit all 11 zinc-dependent HDACs, and we don't necessarily want to do that because it may be associated with unnecessary side effects. So in my research group, we look at ways to make selective HDAC inhibitors for HDAC 1, 2, and 3. And there's quite a novel way by which we do this, making these molecules called PROTACS, or proteolysis targeting chimeras. So these are organic molecules that consist of a protein for the ligand of interest, so highlighted in red. So these are just examples of some PROTACs from the literature. A linker, highlighted in black here, and then an E3 ligand. Don't worry too much about that. That's just something that recruits an E3 ligand to degrade the protein of interest. And the big difference between PROTACs and inhibitors is, well, these are inhibitors. They inhibit the catalytic site. They inhibit the function of the molecule. The PROTAC doesn't just inhibit, but it degrades the protein target. So if you can imagine an inhibitor is like a stone being thrown into a machine to inhibit to stop the cogs working. PROTAC's a bit like, I don't know, maybe a bomb. If you throw a bomb in there to blow up the machinery, it completely degrades the protein, breaks it up into its components. I'm not going into too much detail about this, but basically if we have a HDAC and its complex, we have the PROTAC, we can initiate degradation, regenerate the PROTAC. The research questions we asked is, can we degrade class 1 HDACs using PROTACs? So HDAC 1, 2, and 3. Can we have selectivity for HDAC 1, 2, or 3? And these HDACs exist in multi-protein complexes, and what happens to the multi-protein complexes when HDACs are degraded? And we'd really like to develop complex selective HDAC degraders. So these are some of the examples of PROTACs synthesized in my lab by one of my PhD students. So if you look at these PROTACs 1 and 3, they have short linkers, and they have 2 different E3 ligands. These have longer linkers and 2 different E3 ligands again. And this is the bit in red is the bit that engaged the HDAC enzyme. And this molecule here, specifically with the 12 carbon linker length, degrades HDAC 1, 2, and a little bit 3. So you can see here, don't worry about this too much, but this is HDAC 1 abundance, this is HDAC 2 abundance, HDAC 3 abundance. And you can see with increasing concentrations, we lose HDAC 1 and 2 a little bit. So these molecules are not only inhibiting the HDACs, but they're degrading the HDAC proteins. And these are useful in cancer cells. For rationale for that, this is the HDAC2 enzyme. This is the E3 ligand. You can see the 12-atom linker brings them into close proximity to cause that degradation. But with the 6-atom linker, it's just a slightly bit too short and doesn't do that. So that's a little bit about my research. So I'm involved in synthesis and using these molecules to find new biology and new therapeutics, hopefully. The other researchers, so Richard Dovston is involved in stabilizing protein-protein interactions. So there are estimated to be over 300,000 protein-protein interactions in humans alone. And there are some protein-protein interaction inhibitors, but not really many protein-protein interaction stabilizers. And that is what Dr. Richard Dovston's group is involved in, is trying to develop new molecules that can stabilize protein-protein interactions. And these could also be important in disease, because you could imagine there could be some disease areas where actually stabilizing a protein-protein interaction is more beneficial than inhibiting that. And that's what Dr. Dovston is trying to do, develop new protein-protein interaction stabilizers. This is just an example of some of his research. So the protein he works in as a template is called 1433. Some of his researchers, so one of his PhD students, is interested in developing new ligand discovery techniques. So how do we discover new molecules that stabilize protein-protein interactions? Well, this is one research area where they're trying to develop new methods to discover new protein stabilizers. 1433 interacts with another number of different proteins involved in diseases as well. And he's specifically involved in making covalent and isoform specific 1433 stabilizers. So developing small molecules or bigger peptides that can stabilize these. He's interested in looking at ternary complex dynamics. So that's whenever the two proteins come together to make a bigger protein complex. So we can label up the 1433 protein and a peptide partner And using physical techniques with Professor Andrew Hudson, we can look at fret transfer. He's also looking at chimeric modulation, so looking at heterobifunctional molecules, a bit similar to what I was doing with the PROTACS. Other ways 1433 interacts with other proteins, studying that. And also using mass spec techniques to determine how these small molecules can stabilize these protein-protein interactions. It's quite challenging because these protein-protein interactions can often be low affinity, but can still be very, very important. So even if they're not, if they're low affinity binders to stabilize that interaction, they can still be important. Hopkinson, so this is funny. Me and Hopkinson often get confused. So I'm called Hodgkinson, he's called Hopkinson. Often I'll get some of his emails, he'll get some of my emails. For future reference, this is Hopkinson's research, which is different to my research, which is a Hodgkinson group. So he's interested in looking at small molecules in health and disease, particularly a small molecule called formaldehyde. You can find more out about his research here. So he's specifically interested in this very simple molecule here, formaldehyde. Very simple, but very important. So what happens if you're exposed to formaldehyde? If any of us are exposed to formaldehyde, and that can happen because it is around, you can get pain, skin rashes, breathing problems, nausea, convulsion, coma. Chronic exposure, which can happen to people in industry who are working with these, can end up getting cancer or neurodegenerative diseases due to exposure to formaldehyde. And how formaldehyde induces toxicity in disease is unknown. So that's a big part of the Hopkinson group, is trying to use small molecules and other techniques to see how formaldehyde does these things. You know, there are external sources of formaldehyde: air pollution, food, cosmetics, smoking. There are endogenous sources. We naturally make it in the body. Sometimes it's involved in healthy roles in the body. And other times it can be involved in disease and cancer and pain. And the differences between when it's involved in healthy roles and disease is often not very clear. We still don't know a lot about formaldehyde and how it regulates processes in the body. When is it involved in healthy metabolism? When is it involved in disease? And that's a lot of the Hopkinson group research, is trying to determine the role of formaldehyde in the body with different probes. So what they do in there is they make cellular tools to quantify formaldehyde, chemical tools, so small molecule probes, assay development. They profile how formaldehyde reacts with amino acids. They do studies in formaldehyde metabolism using NMR studies, cellular studies, and enzyme assays. And another researcher here in chemical biology is Dr. Philip Ash. He's also present here today. He's interested in metalloenzymes. So these are enzymes that have metals in their enzymatic site to catalyze transformations with small molecules such as carbon monoxide, carbon dioxide, and hydrogen. Again, you can find his website here. So this is Dr. Ash here, and you know, some bacteria can survive using hydrogen as their only source of energy, and carbon dioxide is their only source of carbon. How do they do this? So this is a picture. I think this is a electron microscope image of some bacteria, these long rod bacteria. And they do this by catalysis. You know, in their active site, they have iron cores, nickel metal cores, that can carry out chemical reactions and transformations. In this bacteria, the one particular enzymes he's interested, they take hydrogen, using this enzyme and break it down to 2 protons and two electrons. And this is the exact same reaction that occurs at the anode of a hydrogen fuel cell. So you can understand why this is important. You know, this is generating electrons and energy, and which is obviously incredibly important for all sorts of industrial applications in clean energy. And you can imagine if we can harness the power of enzymes and understand how they use these abundant metals such as nickel and iron to make these electrons and energy, then this could be a really powerful way of making clean energy. So really important to understand these processes, and Dr. Ash uses a number of methods to try and understand these processes. you know, you can hydrogen oxidation, you can turn the catalysis on and off by using a supply of electrons to hydrogenate using electrochemistry. And electrochemistry allows them to shock the enzymes, and they can use high-power lasers and X-rays to record snapshots of hydrogenase in action. And this is important because if we have snapshots of how hydrogenase catalyzes these reactions, then we can understand how they work. We can potentially use these applications in making cleaner energy as well. And thanks for listening. That's just a few research highlights of a couple of this here. There are obviously others there that I haven't talked about, and you can find their research on the website. Yeah, and you know, you can find a lot on the website. We're all very friendly. If you want to email us, fire us an email. I'm happy to talk anytime. And thanks for listening. Hopefully, you can take any questions.
Thank you very much. Does anyone have any questions at all?
Hi. So,
Hi. Yeah.
With regards to the drugs that attack the cancer cells, is it specific to cancer cells, or is it more of like a broad range thing? Because I know chemotherapy is more of a broad range sort of
Yeah,
treatment.
that's a very, very good point. I mean, yeah, I mean, think of cisplatin, which is, you know, been done for a long, long time, still used today. You know, obviously the problem with these things is they hit normal cells as well. We're always aiming towards to try and make cytotoxic compounds towards cancer cells, and I mean, some of them are. But yeah, that is a goal, and it's not something that's easy to achieve. But it's something that we do aim towards. I mean, with the HDACs, they do have some unnecessary side effects, but I guess it depends which enzymes involved in which cancer in which cell, and it's not easy, as I'm sure as you well know. It's very complicated. Mainly, we look to just get some molecules that can kill some colon cancer cells, but obviously it's a very long process. You know, if it kills cancer cells in a Petri dish, well, it doesn't necessarily mean it's going to, you know, get to the colon in the body. So it's obviously really preliminary stuff. I guess the main aim for us would to try and hit cancer cells, and hopefully with selectivity, and then after that, it kind of would be taken on more somewhere else in terms of drug discovery.
Mm-hmm.
Yeah.
Thank you.
I have a question for James.
Yep.
Yeah. In the design of your product.
Yep.
So how much importance do you give to the linker?
Yeah, the linker is really important. If it's too short, it has to make a protein-protein interaction. If it's too short, then obviously there's going to be steric clash. If it's too long, obviously the protein-protein interaction is not going to be effective. And it gets even more complicated actually, because you know the linker also affects the hydrophobicity.
Yeah, that's what I was thinking.
Yeah, that affects what gets in and what gets in the cells. And to be honest, we're at a point now where we don't know sometimes, is it due to the linker length or is it due to the hydrophobicity of getting into the cell? And sometimes I think actually it's hydrophobicity. Yep.
So I guess though you do have potential for varying the linker, not just for the length, to like functionalize it differently.
Yeah, definitely. Yeah. The problem is there's just too many options. That's the problem.
Need more students. Come on.
Yeah.
Who wants to do an MChem with James?
Yeah.
Yeah, thanks, James.
No worries.
Does anyone have any other questions or comments they might like to make? I'll take the silence as a no. So thank you very much, James, for speaking today.
No problem.
Up next, we have Dr. Dylan Williams, and he's going to be talking with us about some of the learning enhancement that he's doing at Leicester.
Thanks very much, Rosie. Right, I just try to share the right. It's really confusing this. I've got two conference presentations open on my laptop. I don't want to open the wrong one. So I believe, hopefully we can see this one. Everybody see it?
Yeah.
Fantastic. Okay, good afternoon, everyone. So like Rosie just said, I'm going to be talking about what we do in the Chemical Learning Enhancement and Pedagogy group, because it's quite different to some of the other research groups that we've heard about today. So I'll start with a little bit of introduction on what we do, why we do it, and how we differ from some of the other groups. And then I'll give some examples of some of the work we've done and how it's impacted on the way we teach chemistry here at Leicester, and in some cases beyond Leicester. So the group is essentially myself and Richard Blackburn. So there's two of us who are kind of behind everything that we talk about today, really. And there's a few other people who've helped out, who've made some contributions, but it's Richard and I that have done the majority of this. And it's quite interesting to note that neither Richard or myself were trained as educational researchers. So neither of us did PhDs in educational research. We both did what you might call traditional chemistry PhDs. So Richard's PhD is in chemical biology, my PhD is in nanotechnology. So very much experimental laboratory-based chemistry. We both made the transition to chemistry education after finishing our PhDs. It was both something that happened around the same career stage for both of us, although Richard, as I'm sure he will remind you on many occasions, is a lot younger than me, many years after. So one of the things that we often ask is, why do we study chemistry education? Why do we do research into chemistry education? Because obviously chemistry has been taught at degree level for a very long time. So surely we know all there is to know about how to educate people in the subject, and we don't need to do any further research. Well, that's not necessarily the case. That's not really the case. That's because there's an ever-increasing range of new challenges that crop up very, very frequently because of the changing needs of students, the changing needs of graduates, so the kind of skill sets they need when they go out and get a job after they've finished doing their degree. That's constantly evolving, constantly changing to meet societal challenges, for example. The technologies that are available to us to help support education, those are changing at a phenomenal rate. So we need to keep up with that. We need to make sure we're using the best technology and we're using it in a smart way that will actually help our students learn and help our students benefit from those experiences. So I've kind of summarized a few key things, that to me are the most important reasons we do chemistry. So the first one is overcoming misconceptions. I think to explain that one, you've got to think about what is chemistry. And chemistry is about learning how to think about things that we can't actually see. So it's learning how to develop mental models that allow us to understand processes that occur on a size scale where it's impossible for us to directly visualize them, to directly see them. And if you think about the types of models we create, we start from day one of university education. Basically, when you work out how to determine the shape of a molecule, that's a model. When you think about organic reactivity in terms of curly arrows and electron pushing, that's a model. Now, a lot of these models are taught pre-university. A lot of them are taught at GCSE stage or at A-level stage, and they can be very, very challenging for students to understand. So they can be quite abstract, quite unusual, difficult to relate to past experiences. And sometimes that means people form an incorrect mental picture of the process that they're thinking about. So they form a mental model that works for them, seems to make sense to them, but it doesn't actually correspond to the widely held model that is used by everyone else. And we refer to that as a misconception. And it can actually be quite difficult identifying those misconceptions, but it's important that we do, and we challenge them, and we help students overcome the barrier of correcting that initial misconception. We want to inspire future students. We want to think about what can we do to make students proud to be chemists? What can we do to make students excited to be studying the subject, to want to pursue a career in chemistry? What are the steps that we can take to help ensure that we do that? We want to be able to develop the kind of graduate attributes or the graduate skills that are needed by employers And the skill sets that are needed by employers now, the things that they expect chemistry graduates to have now, that might look very different to how, compared to how it did 10 years ago, 20 years ago, 50 years ago. That's a constantly evolving picture. So it's really exciting, but it means there's a big challenge kind of keeping up with that and doing the research to ensure that we're still delivering the experience that allows students to develop those skills. We want to make chemistry inclusive for all. So we want to break down some of the traditional teaching approaches, which are very much focused on a particular type of student with a very particular background. We want to deliver chemistry learning experiences that everyone can relate to. We want all sections of the community to be fully represented in university chemistry and in professional chemistry. So we want people working in the subject as well after they graduate that represent all sections of the population. Like I said earlier, the last thing I've put here, the last of my big priorities is to make sure that we make smart use of technology. Not just think, oh, that looks cool, let's try that out. To actually research it thoroughly, find out how to use it most effectively, and then implement it in a logical, reasoned way. So in terms of what the kind of driving force, the driving factors for research and innovation in the subject, that last 20 years or so, I've seen massive changes to student recruitment. So the backgrounds, the pre-university qualifications, the pre-university experiences of students who arrive at university now are very different to 20 years ago or 30 years ago or 40 years ago. And in some cases, in some UK institutions, the way that chemistry is taught hasn't really changed very much over that period of time. So it's important that we do research that helps us develop a curriculum that matches that past experience and those expectations of students as they arrive in university. There's been an emergence of what we call a graduate skills gap. So the graduate skills gap is a gap between the skills that students leave university with and the skills that they actually need to be competitive in the job market. So there's been this gap in terms of what we were doing in university, what kind of skills training we were giving, and what students were leaving with and what they needed. So that has been another big driver for changing the way that we teach, doing research on how to do things more effectively. The last key thing that I'll put on here, this is based on quite a comprehensive paper which summarized lots and lots of individual studies. There is quite conclusive evidence that proves the traditional method of teaching, which formally we call that didactic teaching. So when you get lots of people just doing a lecture to you, and you can imagine having a 9 to 5 day of lectures one after the other, that's a very traditional way of teaching a degree subject. There is now an overwhelming body of evidence to state that that is not an effective way of teaching chemists or science students in general, for that matter. And that might seem obvious. You might think, okay, well, it's a terrible idea to give somebody a full day of lectures on chemistry. But it's actually been quite a difficult lesson for some people to learn in the subject. They've been doing it for so long and they've been relying on lectures so much, it's quite hard for some academics to kind of come out of their comfort zone and do things a little bit different, a little bit more innovative, in ways that allow a more rounded skills development experience and a more in-depth understanding of the subject to be developed by students. There's a few really, really good and really important rationales for what we do there. So what do we actually do to respond to those big challenges? In order to help students develop that really well-rounded skill set that I was talking about earlier, we adopt inquiry-based learning approaches. And we did this in a very pragmatic way, and we researched it very carefully as we were introducing it. We worked with our students, we interviewed them, we did questionnaires with them, we observed how they worked on these problems. But we use team-based challenges which are inspired by the kinds of activities that professional chemists engage with. So the challenges have real-world scenarios. They're inspired by things that professional chemists are working on, and the students have to work together in groups to solve those. And those typically run on for several weeks. You can think of them as kind of mini projects that are meant to kind of mimic the workplace. And those help students to apply their understanding of the subject in a variety of different ways, helps them to be assessed in a variety of different ways, and to develop a number of different transferable skills. So maybe not necessarily chemical skills, but skills like teamwork, organization, communication, that kind of thing. We've enhanced the pre-lab experience as well. We've done that by using a particular software package that allows us to give our students simulated versions of the techniques that they'll be working on in the laboratory environment. We've long and hard to research the best way of doing that, and I'll talk about that in a little bit more detail in a few minutes. The other thing that we've worked hard on, and this is based on that skills gap that I was talking about earlier, we've worked hard on integrating workplace skills throughout the curriculum. So we embed the development of those key skills, things like teamwork, communication, organization, time management. All of that is kind of implicitly embedded in the chemical curriculum. So we don't say, okay, we're going to stop doing chemistry and we're going to do an hour of skills. We're going to learn how to do a talk, or we're going to learn how to organize our time. What we do, we learn through practice, basically. We learn by setting examples of learning experiences where students have to apply these things, and we give students feedback on how to improve them as they work through. So it's done in a very guided, supported, facilitated way. So I'm going to go back a few years here, and I'm going to reflect on a little bit of research that I did just going back about 4 or 5 years ago, maybe this was. And this was a series of questionnaires, and this is one example of some of the data that I generated from questionnaires I ran with new chemistry students in their opening weeks at university. And I thought this is a great time to ask them, what do you think your degree is going to look like? What do you expect from your degree? And I give them various different kinds of learning experiences as examples, and I just asked them to state whether they agreed they thought they would be part of the degree, or whether they disagreed they thought they wouldn't be part. And actually, the majority of things I asked, 3/4 or more students said, yeah, I think those will be on my degree. And that's a good thing, because actually all of these things should be integrated into a degree. The one thing I would probably question is the first one, memorizing facts and information. Probably don't really need to memorize all that much in chemistry. It's more about understanding the memorization. However, the two in the middle, so opportunities to be creative and opportunities to communicate science verbally, those are the two that got less than 3/4 of the students agreeing with them. So opportunities to be creative, only 50% of the students who responded, 90 students responded, so only 50%, 45 of them in total, agreed that they thought a chemistry degree would allow them to be creative. And that actually horrified me, because when you think about what chemists do professionally, we're coming up with creative solutions to problems. And that ability to think in an innovative, novel, creative way is a really important skill for chemists to develop. The other thing that I was quite surprised by was the next one, which is opportunity to communicate science verbally. There's actually 26% of students responding to that questionnaire who didn't think that was going to be part of their chemistry degree. So it suggested that there was some work that we could do, suggested that there were some things that we could do to try and challenge these almost negative expectations, if you like. And this is where the inquiry-based learning approaches come in. And we call the specific type of inquiry-based learning we use at Leicester is called context and problem-based learning, which for obvious reasons, because it's clumsy to say it every time we call it CPBL instead of yours. We've been working on CPBL now since 2007, and I don't sound too big-headed, but we're good at it. We're looked upon as a bit of an authority on CPBL in chemistry by the rest of the university sector in the UK. That's quite a good thing, because we're thought of as being the experts, and people come to us for advice and guidance, and sometimes to ask us to help them deliver their own CPBL. The basis of the CPBL is these many projects that we design in partnership with industry partners. So we get people who work in the private sector to help us design these problems. And we get students to work on applying their understanding in professional context. For example, our second-year students work on a challenge based on sustainable energy. They're advising a small European nation on how to make informed decisions on its sustainable energy strategy for the next 100 years. And when we do CPBL, when we do these team challenges, we build these kind of core values that you can see on the right-hand side of this slide. So we've got communicating science and doing that in a variety of different ways, verbally, in written form, in poster form, online, all of these different ways. We naturally embed that into these problems. Working with others, well, all of this is done in teams. All of these activities are based on teamwork. Problem solving and creativity. So a lot of these things require thinking outside the box. It's not just, oh, apply what you learned about in lecture 12 and you'll be able to answer this. A lot of it is, okay, you've got to think really creatively, really dynamically. You've got to piece together lots of different information sources to be able to come up with a viable solution. And the important thing is, these types of problems, they don't normally have just one right answer. They have multiple viable solutions. It's just how you come to it and how you justify it. We also use this as a good way of raising business and industry awareness. So it's a good way of enriching the curriculum with how professional chemists use their understanding of the subject. So in terms of how it works, it's a cyclic strategy. Students work on this kind of planning, research, and development cycle that you can see on the right-hand side of the screen. So they start by filling out a template problem plan that we give them. It sounds really formal. It's actually just a piece of paper with a T written on it. But there you go. It's quite. We do a little bit of training in terms of how to fill that out. Once that initial plan has been done, the groups go away and they research the relevant parts of the problem. So they might divide up the task amongst the individual members. Each person might have to go away and research a different aspect. They then need to come back together and start working on developing a solution by bringing that research back together as a team. Once they've got a draft solution, they reflect on it and they think about, okay, well, is this a viable solution? Is this actually addressing the needs of what we're meant to do? And then there will always be room for improvement. So the next part is to go round that cycle again and continue kind of iterating the solution that they've come up with. Oh, hello, that's frozen for some reason, but there you go, we've moved on now. So just one little example of how we do this. We actually have a CPBL induction activity just to get students used to the general features of the CPBL approach. This is called Learn on the Move. It takes place at the start of the degree program. Under normal circumstances, it's a half semester long activity. This year, because of COVID and because of remote learning, we did it as a full semester long activity. And it's based on the design, development, and evaluation of a learning resource. And the idea is students are working in teams to develop a novel learning resource that can be shared with the other students on the course. And it should work in a format that it's portable enough that it could be used on a 20-minute bus commute from the halls of residence onto campus. Part of the assessment of it involves doing a Dragon's Den style pitch. That's the opportunity we use to give students feedback on their ideas. And the final assessment is the actual learning resource that actually gets handed in. And we've had loads of really great examples of very innovative, creative resources there, including games and videos and podcasts, all sorts of really fantastic creative outputs. And we've actually, we've published the actual learning experience. We published the Learn on the Move activity a few years ago as an example of an induction activity for CPBL. But actually, one of the learning resources that was generated was a card game. First year we did it, we got a card game called Go Fisher, which is an organic chemistry card game to help students learn how to name organic molecules and identify organic structures. And that has also been published. That was published as a student-led output. So it's not just that we think it's great, it's that the entire community thought that this thing that was generated by a team of Leicester first years in their first 6 weeks at university was an excellent product, and it was good enough to be published in an American Chemical Society journal. So in terms of the impact that has, it gives students a great opportunity to develop some of those transferable skills, like developing their planning skills, discussing scientific topics with their peers, with other students, reinforcing their existing knowledge. It's also really good in terms of the social benefits, making new friends. This is an induction activity. a large reason we do it is to get students together and get them to fit in at a very early stage. It's really, really good at that. One thing it's not quite so good at, the second category on there, is discuss science with the facilitator. The facilitator is the member of staff or postgraduate student who's helping that group along the way. It's not necessarily a bad thing, that though, because most of the discussion should be student to student rather than student to staff. So the next example I'm going to show you, this is actually the last example I'm going to show you, is what we did to reinvent the laboratory experience using technology. So again, we thought about the laboratory as a holistic process, and that process involves some elements that take place outside of the lab, some that take place in the lab. So preparation, the first part, that is largely done outside of the lab environment. The experiment, that is obviously done in the lab environment. The analysis, some of that may be done in the lab environment, but there may be some scope in some experiments for some of that to be done at home. And then the write-up is typically done at home. So how do we bring these different elements together as a cohesive process to support students through them? And I think one of the things that we were particularly concerned about are these first 2 points. So the green sections there, they relate to that preparation. They relate to what students did at home before coming to the lab. We gave them a manual, we gave them a booklet, and that booklet was given in printed form, and it was also available online, exactly the same thing online, just if you prefer, you can watch it there, you can view it there. And it had information on the safety of the experiment, it had the procedure of the experiment, had a little bit of scientific background on each experiment in it. So give students that, obviously, makes sense. We also got students to do pre-lab quizzes, get them to basically to make sure students were reading the manual. The questions about the stuff in the manual, basically. So a little bit about the science of the experiment and a little bit about the safety of the experiment normally. However, we were concerned that we were missing an opportunity to enhance that preparation element. So you can see those 2 blue boxes we got beneath that. That's something that we introduced 4, maybe 5 years ago. So we introduced instructional videos where we showed students how to set their experiments up in a video that they could watch as many times as they like before coming to the lab. We also have this simulated technique or simulated experiment, which is a third-party piece of software we bought in, and we did some research on its effectiveness. And then that preparation stage was finished with a demonstrator briefing, which takes place at the start of the lab session before students do the experiment itself. So the pre-laboratory simulations, you can see a little animation of one of them here. Some of you will have had a chance to have a play about with these in your lab sessions already. These give students the opportunity to engage with the technique that they're doing on the day. So they can set the apparatus up virtually, they can change a lot of different parameters, and if there's a problem, like you can just see there, you get some feedback straight away. It tells you this isn't going to work, vacuum's turned off. So it gives you feedback immediately, telling you what has gone wrong and how to fix it. You can then have another go, fix the issues with your first attempt, press check again, and if you've got it right, you get that green box that tells you correct, you've done it okay. You can do this as many times as you like, and you can do it, you can make mistakes intentionally to learn why they're mistakes, because there's detailed feedback on everything that you do. So it's really valuable, even if you go through it just to kind of deconstruct it and see what all the mistakes, all the possible mistakes are. When we researched this, when we looked at this a few years ago, we found the students made phenomenal use of this. We initially just bought it on a trial license. So we had it for something like 6 months. We had it for 1 full term. And over the course of that term, it was viewed over 4,000 times. There was an average of 5.1 views per week per student that we made it available to. That is phenomenal. That massively exceeded our expectations. We interviewed the students and we got them to do some questionnaires based on their engagement with this tool. Students told us that they liked using the simulations for about 10 minutes, maybe up to 20 minutes at most, typically on the day before they went in the lab. It gave them a nice kind of debrief on the kind of technique that they'd be using and how to engage with that equipment. The average number of views per student was actually a little bit higher for students who self-identify as having a learning difficulty. So that was a really important finding for us, because again, that addresses that accessibility challenge, or it helps to address that accessibility challenge. It makes the subject more accessible. It makes learning easier to engage with for all of our students, which is really, really critical, really at the core of what we're doing. So on the right-hand side, you can see some data. You can see for the first years who have their labs on Wednesday mornings, the most popular day to view the simulations was on Tuesday, the day before, as we expected. Some students, did it on the morning of the lab, and why not? If you're on the bus and you've got means to do it on the way into campus, then you could have a little refresher, or while you're waiting downstairs in the reception, there's an opportunity to do it there as well. And then, of course, some devotees were even doing it over the weekend, which is good to see, interesting to see. In terms of the impact it had on student learning, in the questionnaire, we found that it had a positive impact on students' ability to set up the experiment correctly. So you can see we gave the questionnaire before we introduced the simulations, and then we did it again a few months after we introduced the simulations, and you can see the percentage who were confident they were able to set up the experiment goes up by 6% there. We also asked whether that pre-lab experience provides suitable preparation. That's also quite a nice increase. So you can get, we got an increase of, I can't see my screen, it's about 10%, I think, isn't it? 59 to 69, I think. So there's a nice increase there. The other thing, it made students more confident. It made them feel more comfortable in the lab environment. So it helped overcome those initial nerves, fear of that unfamiliar environment for the first year students. But it retains its usefulness right into second year as well. And even some of our third year students say that they go back and they review them when needed. Last slide is just some acknowledgements. So Richard, Derek, and Di for some fantastic support here in Leicester. The RSC has funded a lot of the work that we've done. We've got external project partners everywhere, all over the world. McMaster, which is in Canada. Johnny Woodward, who was my mentor, he worked at Leicester when I started working here. He's now in Tokyo. Giro, who's in Freiburg. I think there's one too many I's in that Freiburg. Barbara, who's now at Nottingham, and Kevin, who's our regular industry partner. He works at, he owns actually KKI Associates up in Edinburgh. And of course, most importantly, our students and all of our trial partners. And also thanks to you for sticking around for this late in the day conference talk. And if you got any questions, please fire away.
Thanks, Dylan. I definitely agree that simulations definitely help. I mean, I remember first year when you're going in, you're so nervous about labs and you have no idea what to do. And then, yeah, it's actually quite nice to see how to do it without making things explode in person.
Yeah, I think that was one of our main motivations. It was actually after chatting to some of the first years in must have been about 2015, 2016, and we were having conversations with them, and it was that feeling of intimidation because it's such an unfamiliar environment. It's so different to a school lab. When you do your A levels, you're lucky if there's a fume hood in the room you're doing chemistry in. Sometimes when you're studying your A level, so the university chemistry lab experience is so alien for students coming into university that we just wanted to make it a little bit easier. We just wanted to do something to kind of help with those nerves, help with set some kind of expectation of what it's going to be like before actual arrival in the lab.
Mm-hmm. I have a quick question. I was just wondering whether you think there's a place to teach future chemists through an apprenticeship, or whether you think it should be more university education based.
No, I absolutely agree that there is room for that apprenticeship mechanism. And actually, I don't know if you're aware, but this is something that is gaining quite a bit of traction in the sector. So there are some universities, I think Nottingham is the nearest one to us that does this, but there are some universities that are doing degrees by apprenticeship. So the student is essentially an employee of a company and is studying by DL a lot of the time, distance learning, sorry, a lot of the time. So they're studying a lot of stuff remotely, but they might have some campus days or some campus weeks occasionally. And they do their degree essentially, because it's part-time, they do it over an extended period of time relative to a student who's studying it 100% on campus. But the RSC is very, very keen on that model and is very keen to explore the kind of logistics of running it, but also the impact that it has on students who go through that experience. very new at the moment. I don't know if Nottingham have graduated their first cohort yet, but it is something that I think is a very exciting opportunity for the future.
Yeah, definitely. Does anyone else have any other questions at all?
I have a comment to make. I just want to talk about PBLs as well. Doing that as a first year is actually really, really fun and really interesting. My group actually won last year. I'm still awaiting my prize from you, Dylan.
Keep reminding me. when we're back on campus on September, you can have it, definitely. Yeah.
I'll keep badgering you about it, but no, it's really good, and it's nice to see that all the research you've been doing has really paid off in the Leicester environment of chemistry. It's really interesting to see, especially going through first year and doing all the stuff that you're now mentioning in the lecture is really, really interesting.
Yeah, it's amazing from our perspective as well, because I mean, we get a lot of benefit from it, because we get to, I mean, doing things like PBL, they're the most fun part of the job for me. They're the most engaging part of the job for me, because it's so, you get to see student creativity at its best in PBL, I think, which is fantastic. The other thing that's been really good is actually watching other universities adopt our approaches as well. That's been amazing, because I didn't ever think that would happen, but seeing people take on some of the stuff that we've published, some of the things that we've gone out and presented externally, that's been fantastic. That has been absolutely incredible. By the way, it wasn't phenomenally popular when we first started doing it, but that's because it looked very different to how it does now. So the first time we ran PBL in 2007, we nearly had to stop doing it halfway through the term. It was so unpopular. So it was, the scenario we used was the students were working as advisors to a Hollywood movie making a sci-fi film who needed some chemistry expertise. And it was so wacky and so out there and so far-fetched that students just couldn't really they couldn't make any connections or associate with it at all. And I don't blame them, but looking back on it. But the other problem with it was there was if I remember right, there was an assessment virtually every week. So they had to write a written report pretty much every week back to the studio. And that's just too much. There's too much stuff to do there. We got it right eventually, though. That's good. You only learn through mistakes like that.
Logan in the chat has just said that he thinks it's a really good system. It will help him get more comfortable with getting back into a lab. Yeah.
That's brilliant. Yeah, I think especially now coming back from the pandemic, and we've had so much remote learning, we've had, you know, some reduction in lab contact time, especially for new students who are coming in from outside of Leicester. I know that that's going to be a huge thing, because, you know, students who've been doing their A-levels, they might not have had very much opportunity to do lab in recent times. So I think anything that we can do to support, that's going to be a good thing.
Does anyone have any other questions they might like to ask?
Dalia, I think you're on mute. Well, at least your microphone isn't.
Okay, is that better?
Yeah.
Okay, I was going to say, having just graduated, I still remember having the first year and going through all of the simulations, and they were very, very useful before going into the lab, and they still would be now if I was able to do those before going into the lab now. They'd still be very useful.
Yeah, it's amazing, and we're actually doing we're doing a follow-up project on it at the moment, which is we're actually comparing our initial findings from a few years ago with another, a similar introduction of that technology, but on the other side of the world in Australia. So we're doing a comparative study with them, and we're hoping to publish that later in the year. So that'd be really cool. But it has been incredibly popular in Australia as well, where we've moved on to doing it now. Unfortunately, because of travel restrictions, we haven't been able to go out there and see. Yeah, I thought that was going to be one of the perks of the project. We get a trip to Sydney, but no, sadly not.
Oh, Dylan, I have a quick question actually. So how often do you re-interview students throughout the course? So even asking the same questions between year 1, year 2, year 3, year 4, and see how the response changes.
So there was a time when, and I guess every one of our current students or recent students who's online at the moment can tell you that I still do this. I send out too many questionnaires each year, and I'm conscious of that. I feel incredibly guilty every time I do that.
You just love the data.
I was doing a systematic study for a while. I was doing a skills awareness study, which was skills perception study. So the idea behind that one was I was asking students how important, so basically to rank an importance for all of these different skills. Some of them were subject-based, so some of them were very chemical skills, others were transferable, others were maybe computational. I had this really long list and you had to kind of score all of these skills. And I asked every year group, every year for about 3 years. And I think by the end of that study, there were some students who were ready to kill me. They'd seen that questionnaire so many times. So I was glad to retire that one. I was glad to get rid of that one because I was like, oh, I'm not doing that one anymore, but I've got a new one.
Okay, I didn't want to bring this up. I have no knowledge about this. I didn't want to cause any pain. Sorry.
I probably do still do it. Send out too many questionnaires each year. What I will say is the student response is phenomenal because it's always optional, obviously, but we always get really good responses, and I can see why we get good responses because it's it's in the long run it's in students' best interest because their responses help us improve the program. But I'm always amazed by it when I look at it and I think, wow, we've got so many responses to this questionnaire. I'm always really thankful, but I'm always amazed by how constructive and how willing to get engaged in all of this everyone is.
That's the reason why I asked was I was interested in how the students' opinion evolve throughout their degree, because at the start they might literally not be very knowledgeable about the degree to begin with, so they have some expectation. They change in year 1, in year 2, year 3, then they see more of the maybe subjects they prefer, and then all of a sudden the opinion just changes completely. Then they go do an industry and say, oh God, I hate everything I've done so far. I just want to go in an industry setting and forget about all the computational chemistry that I did. Let's say, so it'd be interesting to know how their opinion evolves for the degree.
We did, when I did the one that I presented the results from the first years, the expectations, I did do a version of that with the second years and the third years, and I had intended to keep running that for a few years, but I didn't. I kind of ditched that after because I think I felt like I got what I needed out of it, and I thought that's it. I'm going to focus on something. I do have a tendency to do that sometimes. I get excited about something new and move on to it sometimes. But yeah, we got some nice stuff out of it. We got a paper out of it, so I kind of moved on a little bit, unfortunately. But yeah, the skills perception one that I said that I ran for a few years, we got a lot of stuff in there, and it's really, really interesting to see how that evolves over the course of the program. Because what you tend to see is people start rating the kind of workplace skills, so the less chemical facing things, the ability to give a presentation, ability to write a report, work in a team, organize your time. The importance of those things goes up steadily as you work your way through the program. The importance of all the other things kind of slowly decreases, although the phys chem seems to suffer more than the others for some reason. I don't know what it is, but the more phys chem skills seem to decrease at a faster rate than the organic and inorganic. There you go.
Thanks. Thank you.
Yeah, it's definitely on behalf of students, I think it's definitely encouraging when you send out surveys, because it actually shows that you are making the most of our 9 and a bit grand each year, and actually trying to make it the best teaching environment.
Yeah, I mean, we've always been proud of that, and I'll still stick with that we do have the best degree in the country. I think we got the best chemistry degree anyway. And I go to places and I see things that are good in other places, but I've never seen something that looks like such a good package as as what we put on here in Leicester. And I think that's because we're kind of relentless in that way. We never kind of rest on our laurels. We never think, oh, that's it now, it's good enough now. We're always trying to improve it. We're always trying to do a bit more work on it. And I think it's rewarding to us, and it's rewarding to all of you guys as well, which is amazing.
Okay, does anyone have anything else they might like to say, or any other comments? Another big silence. I'll take that as a no. Thank you everyone for coming, and thank you Dylan for speaking today. Everyone is free to go, but Callum and I will stay around for another few minutes if anyone here is a prospective student or is coming to study chemistry or any other subject, and we're here to answer any questions. So thank you everyone.